Microfluidic mixing techniques for precise reaction control represent a transformative advancement in both chemical and biological research, enabling scientists to execute reactions with an unprecedented level of precision, speed, and reproducibility. In these systems, fluids are manipulated within channels that are typically tens to hundreds of micrometers wide, where flow is predominantly laminar rather than turbulent. This controlled, predictable environment is essential for achieving uniform mixing and controlled reaction kinetics, which are critical for applications ranging from chemical synthesis and drug discovery to diagnostics and cellular biology. To truly appreciate the advantages of microfluidic mixing, consider the analogy of a traditional large-scale reactor. In a conventional laboratory setting, reagents are often combined in a beaker or flask and mixed using a stir bar or mechanical agitation. However, in such bulk systems, mixing is uneven and can be highly variable, akin to trying to mix a thick batter in a huge bowl with a wooden spoon. Temperature gradients, localized concentration differences, and inefficient heat transfer can lead to inconsistent reactions and poor reproducibility. In contrast, microfluidic devices function like a state-of-the-art miniature factory where every fluid element is precisely controlled and mixed uniformly. Imagine a high-precision assembly line where each component is handled with the same meticulous care, ensuring that every mini reaction on the chip is identical to the next. One of the most popular techniques in microfluidic mixing is chaotic advection. This method relies on the design of intricate channel geometries that force the fluid streams to split, stretch, and fold repeatedly as they traverse the channel. A prime example of this is the staggered herringbone mixer, SHM, where tiny grooves are etched onto the floor of the channel. These grooves induce swirling motions that cause the fluid layers to interlace and merge continuously. Think of it as a river flowing over a series of rapids and bends, where the water is forced to twist and turn, ensuring that the colors of different streams mix uniformly. Chaotic advection is particularly effective because it overcomes the limitations of diffusion alone, especially important in microchannels where molecular diffusion is often slow relative to the fluid flow. Another method is lamination mixing, where two or more fluid streams are forced into extremely thin layers, or laminae, that are stacked together. In this scenario, the large surface-to-volume ratio of the thin layers facilitates rapid diffusion between them. Picture a lasagna, where layers of pasta, sauce, and cheese are so thin that the flavors meld together almost instantly. In microfluidics, lamination mixing is often achieved using T-junctions, or flow-focusing devices that split the fluid into multiple thin streams before recombining them. This technique is particularly useful for reactions that require precise control of reagent concentrations, or when sensitive biological samples must be mixed gently without applying excessive shear forces. Droplet-based mixing is yet another innovative approach, where reagents are encapsulated within discrete droplets that serve as isolated microreactors. In these systems, droplets are typically generated in an immiscible oil phase, and each droplet acts as a tiny vessel in which mixing occurs rapidly due to internal circulatory flows. Imagine shaking a bottle of salad dressing. The vigorous motion causes the oil and vinegar to emulsify and mix uniformly in microfluidic droplets. The mixing process can be further enhanced by designing channel geometries that induce internal vortices within the droplets, or by applying external stimuli like acoustic or electric fields. This method is ideal when dealing with reactions that must be compartmentalized to prevent cross-contamination, such as single-cell assays or high-throughput chemical screenings. Active mixing techniques further expand the capabilities of microfluidic systems. Unlike passive methods, active mixing employs external forces to promote fluid agitation. Examples include using magnetic fields, acoustic waves, or electric fields to perturb the fluid motion within the channels. For instance, magnetic stirring can be achieved by embedding magnetic nanoparticles within a fluid and then applying an oscillating magnetic field. This approach is akin to using a handheld whisk in a bowl of batter, except that in a microfluidic channel, the energy can be delivered with pinpoint precision and without physical contact, Similarly, acoustic mixers utilize ultrasonic waves to induce cavitation or microstreaming, thereby enhancing mixing even in viscous fluids or at low flow rates. One might ask, what are the main challenges in microfluidic mixing and how do these techniques address them? The primary challenge is that at the microscale, fluids tend to move in parallel layers, laminar flow with minimal natural mixing because turbulence, a key driver of mixing at larger scales, is virtually absent. To overcome this, microfluidic engineers design channel features such as obstacles, grooves, or curved paths that deliberately disrupt the laminar flow and create chaotic or laminated mixing patterns. Additionally, active mixing techniques can be integrated to provide an extra level of agitation where passive methods might fall short, especially when rapid reactions are required or when dealing with high viscosity fluids. Let's explore some concrete examples. In the field of chemical synthesis, microfluidic mixers have been employed to produce nanoparticles with precise size distributions. By controlling the mixing of precursor solutions in a microchannel, researchers can ensure that nucleation and growth processes occur uniformly, resulting in monodisparate particles that are critical for applications in catalysis or electronics. In one study, scientists used a staggered herringbone mixer to produce quantum dots with exceptionally narrow size distributions, directly correlating the efficiency of the mixer with the optical properties of the resulting nanocrystals. In biochemical assays, precise mixing is crucial for enzymatic reactions where reaction kinetics are sensitive to reagent concentration. For example, in microfluidic enzyme assays, two substrates may be introduced into a mixing channel where they rapidly combine to initiate the enzymatic reaction. The uniform mixing ensures that each droplet or reaction volume experiences the same substrate concentration, leading to consistent reaction rates that can be accurately measured using fluorescent reporters. This level of control is essential for applications like drug screening, where subtle differences in enzyme activity must be detected to identify potential therapeutic candidates. Cell culture and single-cell analysis also benefit greatly from advanced microfluidic mixing techniques. In microfluidic devices designed for cell-based assays, gentle yet efficient mixing ensures that cells are evenly exposed to growth factors, nutrients, and other signaling molecules. For instance, a microfluidic bioreactor might use lamination mixing to create a controlled gradient of a chemotactic agent, allowing researchers to study how individual cells migrate in response to chemical cues. The ability to precisely control these gradients has significant implications for understanding cancer metastasis, wound healing, and developmental biology. Analogies can help illuminate these complex ideas. Consider the difference between stirring a thick soup with a spoon in a large pot versus using a high-powered blender to achieve a smooth puree. The large pot represents traditional bulk mixing where the stirring process is slow and uneven, leading to pockets of unblended ingredients. In contrast, the high-powered blender, much like a microfluidic mixer, rapidly and uniformly mixes the contents, ensuring that every bite of the puree is consistent. This analogy captures the essence of why microfluidic mixing techniques are so valuable. They enable a level of homogeneity and control that is simply not possible in larger-scale systems. Furthermore, the integration of real-time monitoring systems into microfluidic mixers allows researchers to observe the mixing process as it happens. High-speed cameras, fluorescence imaging, and laser-based detection systems can be used to visualize and quantify mixing efficiency in real time. For example, fluorescent dyes may be introduced into one of the fluid streams, and as the fluids mix, the gradual homogenization of the fluorescent signal can be monitored and quantified. This data not only validates the design of the mixing channel, but also provides valuable feedback that can be used to fine-tune flow rates, channel geometries, and other parameters. Such real-time monitoring is akin to having a digital dashboard in a high-performance car where every variable from speed to engine temperature is continuously monitored and adjusted to maintain peak performance. Multiplexed microfluidic platforms also illustrate the power of precise mixing techniques. In these systems, multiple reagent streams are combined in a controlled manner to perform complex assays that require the simultaneous detection of multiple targets. For instance, a diagnostic device might mix a patient's blood sample with an array of different antibodies in separate microdroplets, each targeting a specific biomarker. The precise mixing ensures that each droplet forms under identical conditions, allowing for accurate quantification of each biomarker. This is similar to a multi-lane highway, where each lane carries vehicles that travel at the same speed, ensuring that the overall traffic flow is smooth and predictable. As we look to the future, the development of microfluidic mixing techniques is expected to benefit from advances in microfabrication and computational modeling. Engineers are continually exploring new channel geometries and materials that can further enhance mixing efficiency and reduce dead volumes. Computational fluid dynamics (CFD) simulations are increasingly used to predict and optimize mixing behavior, enabling rapid prototyping of new designs before they are physically fabricated. These simulations can model complex interactions within the microchannels, allowing researchers to visualize flow patterns and identify potential areas for improvement. In this way, the iterative cycle of design, simulation, fabrication, and testing is accelerated, driving innovation in microfluidic mixing. A common question that arises is, how do these microfluidic mixers handle sensitive biological samples without damaging them? The answer lies in the careful balance between mixing efficiency and gentle handling. While active mixing techniques can introduce high energy into the system, microfluidic devices are designed to operate at low Reynolds numbers, ensuring that the forces exerted on cells or biomolecules remain minimal. Techniques such as staggered herringbone mixing or lamination mixing rely on channel design rather than high-energy agitation, thus preserving the integrity of delicate biological samples. This is crucial when working with live cells, proteins, or nucleic acids, where excessive shear stress could lead to denaturation or cell lysis. In summary, microfluidic mixing techniques for precise reaction control are revolutionizing the way chemical and biological reactions are conducted. Through a combination of advanced channel geometries, both passive and active mixing strategies, and real-time monitoring, these systems offer unparalleled control over reaction conditions at the microscale. Whether it's achieving uniform nanoparticle synthesis, conducting sensitive enzyme assays, or developing multiplex diagnostic devices, the precision and efficiency of microfluidic mixers make them indispensable tools in modern research and industrial applications. The integration of computational modeling, automation, and real-time feedback is pushing the boundaries of what can be achieved, opening up exciting possibilities for future innovations.